Cell division is a process by which a cell divides into the two daughter cells. Parent cell was bigger, it has performed its function, now it will be dividing into the two daughter cells. The cell which is divided is called as the parent cell. The cell into which the parent cell has been converted or divided is called as the daughter cell. Before starting of the process of cell division, a parent cell through it have to pass through a phase called as the interphase. Okay, so when it comes to the cell division, before starting or initiating the conversion, the cell have to pass through a phase which is called as the interphase. Before a cell divides, each of its chromosome is made up of a single chromatid. See, one chromatid, when it comes to the pair or a cross, they were called as the chromosomes. One side of the chromosome is called as chromatid. Before starting a cell division, they would be converting into a single chromatid. During interphase, the chromosomes will be doubled. Let's say they were one, they will become two. If they were four, they will become eight. So it means that of each chromosome, it makes a new chromatid or a new single cells. As a result, each chromosome consists of the two chromatids. Every chromosome is made up of two chromatids, which is called as cross generally. So it have to pass through the cell division. The process of cell division involves two phases. Phase number one is the nuclear division and cytokinesis. Nuclear division is a process in which the nucleus is going to be divided. It is the division or breaking down of the nucleus. First, the nucleus have to be divided. The nucleus starts to pinch inward. It starts to fuse into two daughter cells. And once the nuclear division has been initiated, cytokinesis comes up. Cytokinesis is the process of following up of the cell division, cytoplasm. So cytoplasm is inside a cell, the main protected material, just like in case of humans, we have the DNA somehow. Inside the nucleus, DNA is present. So we actually want that DNA to give instructions to the nucleus. Once nucleus starts pinch inward, once nucleus start dividing, this process of division of nucleus is called as the nuclear division. Cytokinesis is the process of cytoplasm. So once nuclear division has been initiated, it has been completed. After that process, the outer protecting layer, the semi-liquid layer, the cytoplasm starts dividing. So if somebody asks you what the nuclear division is, you should be clear that nuclear division is the process of division of nucleus, and what the cytokinesis is, it is the process of division of cytoplasm. So that is how these two activities goes on. Cell division is of two types, mitosis and meiosis. Mitosis keep going itself for the till the life of the individual ends up. Like our skin is growing, we got a wound, that wound repairing is initiated and completed by the mitosis. Meiosis is when it occurs only when they have to transmit instructions from one generation to the next, just like reproduction. When it comes to the formation of zygote, it is followed by it is made by the formation of zygote is created by the process of meiosis. Otherwise, that healing, that repairing, that growth is done by the mitosis. That's it. So the cell division is divided into the two phases. One is nuclear division and another is cytokinesis. When it comes to the nuclear division, it's the process when nucleus just initiates its division or it divides itself into the two daughter cells so that the nuclear material present in the parent cell could be shared with the daughter cell. That is why the DNA of a skin cell is always matchable to the same dermal material or the same dermal parent cells. Meanwhile, once cell division has been done, it converts the cytoplasm is ready to be divided into the two daughter cells, converted into the two daughter cells. But first, the nuclear division needs to be done. Once it is completed, right after that, cytokinesis will be performed.